It's perfect.
All right, all good? All right, so most of you probably familiar with the High-Oleic trait and why it's important. But what may be less clear is that trying to maintain a very high level of purity throughout all the stages of cultivar development and release has proven to be a little bit of a problem, at least for our programs. This presentation is going to talk about why that happened and what we are going to do to fix it. So Bailey is a historically dominant Virginia type cultivar in the VC region. And even though it's normal oleic, it was still about 13% of the certified acreage in North Carolina last year. Bailey 2 is intended as the high oleic near isogenic replacement for Bailey, created by a 4x backcross of Bailey to a high oleic source. And even with limited seed availability last year, we were up to about 44% of the NC acreage, and with better seed supplies this year, we expect that number to continue to increase, and it should be, a kind of dominant cultivar for the foreseeable future. And the problem is that we have a high oleic cutoff of 74%, so you got to meet that in order to be considered high oleic. And Bailey is at 74.4 here in this data, which is from 3 years of our PVQE, which is the regional variety test, is a composite mean. And it's just way too close, and we really want to know why it is coming so close to this threshold, especially because we don't see it in our other leading high oleic cultivars, which are Sullivan and Emery, and we also don't see it in our elite breeding lines. We have 4 shown here. At the time of this start of this experiment, N14023 was a breeding line. It has since been released last year as the new cultivar NC20. And ideally, we want to see all our varieties more, in the safer range around 80. So our hypothesis, we have a couple of possible explanations for why we're seeing this. The first is that the seed lots are just contaminated with these normal oleic seeds that lack the mutation in FAD2B. And it's kind of easy to see how this could occur at numerous stages of the breeding process, whether that be planting or digging, harvesting, shelling, or even in the phenotyping. And also maybe there's a little bit of cross-pollination going on. The second hypothesis, and we've kind of had anecdotal reports of this, is that this oleic acid content is somehow influenced by plant maturity. And this can go either one of 2 ways that we were thinking. One is that if the plots are dug too early, you can have a lot of immature seeds in there that have not had enough time to produce sufficient oleic acid. Or on the flip side, if you dig too late, you can get these over-mature seeds that possibly have lost some of this oleic acid to metabolism. You can have some other versions of FAD2 getting turned on, or some other enzyme that's converting some of that oleic to linoleic or some other compound. The third is that we have these minor modifiers that are segregating in our material, and if those happen to break the wrong way in the segregation, then you can wind up with a line that's suboptimal in terms of oleic acid. And the fourth is that we're assuming that FAD2A is fixed in our program and has been for a long time, and we have evidence to support that. So we're really just looking at this qualitative single gene trait of FAD2B, and so because of that, we expect environment, both location and year, to have no effect, or at least very, very little. So the field experiment is we took the 8 genotypes that were listed on the earlier slide 2 of this presentation. We did this for 2 years, 2019 and 2020, and we also have it again this year. One location, which is just our main location, the Peanut Belt Research Station in Lewiston, and then we did 3 dig dates. So we did what's optimum for us, which is 150 days after planting. We did 2 weeks before that in order to try and catch some of those immature seeds. And then we did 2 weeks after that in order to catch some of those over mature seeds to see what the effect there was. And because of the dig dates, we had to do a strip plot design where any plot that needed to be dug on the same day had to be in the same strip. So we did 2 reps. So that brings us to 96 total plots. So for the phenotyping, we did 96 seeds from each plot. So that brings us to 9,216 total seeds. And we ran these on our Seedmeister. This is the same machine that our program has historically used for oleic acid content. And it's also the same exact machine used by foundation seed producers in North Carolina to verify these lots as high oleic. And how it works is it transmits light to a seed, measures how that light is reflected, and then uses that in a model previously determined by GC to predict the oleic acid content. And I'll emphasize model and prediction, because sometimes it leads to wonky results where you see a seed above 100% or seeds that seem to be way too low down in the 20s or even the teens. And the main drawback to this machine is that it is very, very slow. It takes one person about 30 to 45 minutes to scan these 96 seeds. You go a little bit faster if you're not trying to keep the seeds separate. And so you do the math there, that's 48 to 72 hours just for the phenotyping on this experiment. And a lot of that depends on how this machine chooses to behave on any given day. So for the genotyping, we did do all 9,216 seeds individually. In order to do this, we used the seed chipper and then the crude DNA isolation protocol that was the focus of my presentation last year. Probably a little bit of overkill with the genotyping, but kind of like a secondary objective of this was to really put this seed chipper to the test and see how it would hold up under a very high throughput experiment. So we just set our PCR. We do the PCR, it's about a 2.5 hour protocol here in the thermocycler. I use PACE, which is a much cheaper generic version of the more popular CASP. And the 2.0 version of this is optimized for use for crude DNA. I very highly recommend PACE. Anybody looking to do SNP genotyping. Read the PCR on a plate reader once it's done. And then the analysis is just a very simple R script and a few Excel templates. And that's very quick and easy to do. So here's the primers that we used. Just like CASP, PACE is a 3-primer PCR. Down here, we have the normal oleic sequence and then the high-oleic sequence. And the only difference between them is this single base insertion and the coding sequence with the red A here. And that's what causes your frame shift and the loss of function of your enzyme. And so this assay is directly targeting that causal mutation, which is always nice when you can get that to happen. The common primer is in purple, and it binds to both the normal and high-oleic sequence, and it binds upstream of that SNP. And then you have your 2 allele-specific primers. They're identical in black, and the black part is what's actually going to bind the peanut DNA. They differ in these 5' tails, the green and the blue. This is what's actually going to attract your fluorophore and be read by the plate reader. And these are pretty important sequences to have. You can just slap these on the front of any primer and turn it into either a PACE or a CASP marker. And then they obviously differ in the very last base of the primer, which is what's actually going to interrogate the SNP that you're going after. And you can do a biallelic indel like this just as easy as you can your kind of standard biallelic primer or biallelic SNP. So the normal oleic primer will only bind to normal oleic DNA and amplify, and then your high-oleic primer will only bind to high-oleic DNA and amplify there. So the overall results, it's very high success rate at 98.4%. I consider it's success when you have a high quality genotype call for that individual seed. Did see kind of a nice bump in the second year. I attribute that somewhat to being more familiar with the protocol, but also we switched over to like a bottled molecular biology grade water rather than using tap water coming into the building through a filtration system. And I think that is kind of more of just like an indictment of the building that I happen to work in than anything else. Heterozygosity was 1.1%, no real effect by year. This was surprisingly high to me, especially because we were working with very highly inbred material, so any residual heterozygosity seems unlikely. Cross-pollination is one excuse, another excuse here potentially. I was kind of skeptical about this, and then I saw these pictures from David Jordan of these honeybees foraging in our peanut fields in Clayton, in Lewiston. And I still think it's unlikely it'll cause this level of heterozygosity, but I guess the honeybees are always out there, and they're always going to be hungry and looking for pollen as well. So I think the most likely explanation here is that these are just genotyping errors. Anytime you try and do something really fast and really cheap, I think you're just going to get a little bit of splash on those plates, and whether that's a little bit of the seed chip or it's a little bit of liquid just hopping between wells, I really think that's what's going on here. So we just throw out all the fails and the heterozygotes, and we still wind up with almost 9,000 seeds with both a good genotype and a good phenotype. So here's probably one of the most important slides of the presentation. As you can see in the second column here, none of our lots were completely pure. Some of our lines did have very low levels of contamination, such as N14023 and Sullivan in the green boxes. And we can live with that level of contamination, but some of them were just unacceptably high. We had Bailey 2 at almost 20%, and then N15041 up at 16%. And you can see the means separate nicely as you would expect in the normal versus the high-oleic genotype seeds. So that's a great indication that everything was working as we wanted it to. And kind of not made clear from this is we don't really think that this contamination issue is endemic to any of these genotypes. It's more of probably variation based on seed source, or maybe even just like a sampling error within that large bag of seed that we use to plant these. And that if you were to like repeat the experiment with different seed sources, you may see some changes in what was contaminated. So in a redo of this experiment, you might see Bailey 2 was pure, but Sullivan was contaminated, for instance. And so to kind of look at those other hypotheses I talked about earlier, the next 3 slides, we're just going to be looking at the 7 high-oleic lines. We'll just drop Bailey for now, and just looking at the confirmed genotypically high-oleic seeds in each of those genotypes. So here we're looking at the minor modifiers. 2 big takeaways from the slide. First is that if you're looking at just pure lots of all these lines, they all comfortably meet the benchmark. So that's very good news for us. And the second is that we did see, significant presence of some minor modifiers, and that they may explain, 5 to 5.5% of the variation, from about 79% up to 85.5%. And so that kind of leads us to this open question that we have now: is it worth it to breed for these? And I think my quick answer to that is it depends on how good we are able to get rid of this contamination. If we can do a good job of getting rid of it, it's probably not worth it. But if we're going to have to live with a little bit of contamination, then I think it might be worth it to build in this little buffer here for us. Year was insignificant, which is great news, so that we don't have to worry about the environment playing a role here. And dig date was also insignificant. So that's another thing. You don't have to worry about getting your digs, perfectly right in order to, get that oleic premium. You did see a slight numeric increase with the digs, but that was nowhere near statistically significant. And then, so kind of the whole point of this presentation is, why is this contamination so bad? And I'll try and answer with this slide here. So here in the red, we have just Bailey seeds that were genotyped as normal oleic. And then you have the distribution of their phenotype. And then in the gray, we have just the Bailey 2 seeds that were genotyped as high oleic and the distribution of their phenotype. And the problem is you have this significant overlap between the 2 peaks. And when you're applying a selection threshold of 74%, that's going to create 2 problems. One is you're going to take all these truly high oleic seeds and you're going to throw them out. And that's not a huge problem because we have so much seed. But the problem is when you go to the right of this black line, any of these red that's left of this line is going to be a normal oleic seed that you advance because you think it's high oleic. And if you were to plant out that seed, it grows into a healthy peanut plant, produces 100+ seeds. It's kind of easy to see how that problem can snowball from there. So there's 2 ways we think we can fix this. One is to get a more accurate machine or model that can tighten up the width of these peaks here and reduce the amount of overlap. And the second is to kick that threshold out. And this number isn't set in stone, but for today I'll just throw out 83 as a new cutoff. But using this will create 2 problems. One, you're going to be throwing out a lot more seed. Again, not terribly concerned with this because we generally produce way more seed than we actually use. But the other problem is that there is no way the Seedmeister can scan this much seed because it's such a slow machine. And so how do we solve the problem? Throw a bunch of money at it, and with the help of Foundation Seed, we're going to go out and buy a Quality Sense Q-Sorter Explorer. This hopefully should be on campus by about September. This is a company based out of Switzerland. I think most people should be familiar with them. They probably contacted you about something. And it can process anywhere from 30 to 3,000 seeds per second. Peanuts probably on the low end of this because of the seed size, but this should easily allow us to bump that threshold up to about 83%. We also, with the new machine, hope that it's a little bit more accurate, and we've also worked with them to improve and modernize our grading. So all our grading is going to move to this machine as well. And then as the shift to high oleic nears completion, I think the opportunities for contamination and cross-pollination will continue to decrease just because that normal oleic allele is going to become that much less frequent in the breeding program. And so with that, I'll close with some acknowledgements. Just like to thank NC Peanut Growers, Foundation Seed, and Certified Seed for funding this research, and also 7 Hampton Farms for indirectly providing me gainful employment. So that's it.
Thank you, Ryan. Are there any questions at this time for Ryan Andres? I guess from our program standpoint, I mean, this has been an ongoing conversation that we've had, particularly within our PBQE group, within our regional group, and then also with Daryl Bowman, who's a part of our NC Foundation Seed Producers group. And so, you know, kind of paying attention to, you know, as we're going through the breeding cycles, it's pretty easy to screen that on a seed meister, but once we get closer to you know, whole or like larger plot increases, it becomes a problem. So I guess first question here from Charles Chen, where are those high-oleic seeds of Bailey coming from, since Bailey is non-high-oleic?
Yeah, it was kind of low, but I think it's just you know, somehow you're getting high-oleic seeds when we grow these things out in, you know, close plots. You know, some seed just gets dragged along and makes its way in there. And then when you scan it, the oleic value was kind of on the low end, and so you just advanced it. I think it's just the same seed contamination issue.
A couple comments here. One from Corley Holbrook, excellent presentation, and then Soraya Bertioli, great talk. Thanks for sharing, Ryan. Other than that, I don't see any other. Oh, one more follow-up question from Corley. Do you have any idea of how much foundation seed will be lost with Q sorter?
I don't, I guess. We haven't really gotten our hands on using it at all. I guess we also don't know how much it's going to improve the accuracy there. I think one thing we've also talked about is that maybe for the different cultivars, you know, depending how those minor modifiers go, that maybe you have different thresholds for different lines. So it may depend or you shift a little bit based on phenotype.
Great. Thank you, Ryan.